Hello everyone, let us discuss on the topic nucleus and development on experimental embryology. In this session, we will discuss about the introduction, role of nucleus and the enucleation experiment on various cells such as nerve cells, sperm cells and acetobularia development and also in sea urchin eggs. Before discussing the role of nucleus, first let us give a small introduction to the nucleus, the control center of cell. The nucleus is a membrane-bound organelle present only in eukaryotic cell and it's completely absent in prokaryotic cells, and it is act as the control center of the cell because it stores the cell's genetic material, which is DNA, in the form of chromatin, and also it regulates the cellular and metabolic activities such as cell division, respiration, excretion, etc. And also it ensures the accurate replication and transmission of genetic information during cell division. And it's surrounded by a protective nuclear envelope, and the nucleus maintains its own environment while enabling selective exchange of material via nuclear pores, and it will pass the genetic material into the cytoplasm for further protein synthesis. And this essential organelle not only safeguards the integrity of genetic material but also the orchestrates and its expressions to support the cell's proper functioning. Coming to next slide, we will deal with the role of nucleus, its genetic regulation. And the nucleus controls the gene expression by activating or silencing the specific genes during the different stages of development, such as mitosis and meiosis, and this ensures that cells differentiated into specialized types like muscles, nerve or skin cells. Next role of nucleus is in cell differentiation. By regulating the production of proteins, the nucleus directs the transformation of unspecialized stem cells into the specialized cells, and it helps to establish the distinct cell identities necessary for the forming tissues and organs. Coming to next slide, we will deal about role of nucleus. First one is growth and division. The nucleus ensures the accurate replication and the distribution of genetic material during the cell division, and it's also a crucial process for embryonic development and tissue repair. And next one is epigenetic modification. The nucleus supports the epigenetic changes that influence the development by controlling the long-term gene patterns. Next one is coordination of developmental signals. The nucleus interprets signal from the environment and adjusts the gene activity to guide the development. Coming to next slide, we will deal about various experiments on which prove that nucleus is important for cell survival. So first one is enucleation experiments on nerve cells. Enucleation means removing of nucleus. So in the first experiment on nerve cells, enucleation experiments on nerve cells help the scientists to understand the critical role of nucleus in maintaining the cellular function and the survival, and how the experiment was done by cell isolation is the first step, where the nerve cells were isolated from the organisms such as amphibians or other model organisms. Mainly from amphibians the cells were taken, and the enucleation process is done by using precise tools such as micropipettes or fine glass needles. Scientists physically remove the nucleus from the nerve cells, and the procedure was done under a microscope in a specialized culture condition to keep up the enucleated cell alive temporarily. And observation period, scientists monitor the enucleated cells to study their behavior and protein production and survival over time. And they observed enucleated nerve cells survived briefly using existing cytoplasmic resources. Without the nucleus, they lose the ability to synthesize new proteins and repair the damage or regenerate, or ultimately leading to cell death. So impact of this research proved that nucleus is essential for the nerve cell survival, as enucleated neurons could not produce new proteins or repair themselves. Coming to next experiment on nerve cells, the same experimental procedures were taken. Instead of nerve cells, sperm cells were taken, and sperm cells were collected from the organisms such as sea urchins, frogs or mammals. And this experiment was done. The sperm cells are collected from organisms such as sea urchins, frogs or mammals, and the enucleation process, the nucleus of the sperm cell was removed using micropipettes or through the chemical treatments that destroyed or extracted the DNA. These procedures left only the sperm cytoplasmic components intact. Fertilization attempt, enucleated sperms were introduced to an egg to observe the outcomes during the fertilization and early embryonic period. The findings were the fertilization without a sperm nucleus, enucleated sperm cells can still penetrate an egg. However, without a nuclear material, they cannot contribute genetic information and prevented the proper embryo development. Role of nucleus in fertilization. The nucleus of the sperm provides half of the genetic material needed for the zygote formation, and it's essential for initiating a development and ensuring the transfer of paternal genetic information. The impact of research is confirmed that sperm nucleus is critical for successful fertilization and zygote formation and help to clarify the complementary roles of the sperms and egg in reproduction. Next experiment is the role of nucleus in acetabulary development. It's a transformational or transplantational experiment. Here, the key functions of nucleus is controls growth, morphogenesis and differentiation, and also contains genetic information such as DNA, which is needed for the protein synthesis and enzyme synthesis and direct species cap formation. This experiment was done by Hammerling's, and so it is known as Hammerling's experiment, and demonstrated the nuclear control of development using two species of acetabularia. Acetabularia mediterranea, which have a round cap, and Acetabularia crenulata, which is a branched cap. Here the nuclear transplantation is done where the cap regeneration matched the species of the nucleus, proving the nuclear control of the cell. Here the regeneration, if the nucleus is present, the cell can regenerate the stalk or cap. Without the nucleus, regeneration does not occur. Interaction of nucleus and cytoplasm. Nucleus produces mRNAs for protein synthesis. Cytoplasm execute the development based on the nuclear instruction. So, nuclear act as the control center of the growth and development, determine the species traits and control the timing of the development. So, without a nucleus, a cell cannot survive. So in this picture it is clearly shown the Acetabularia mediterranea and Acetabularia crenulata. During her studies, Hammerling discovered that Acetabularia, a type of marine algae, had a single nucleus located in the base of the cell, and also the experiment was he successfully transplanted the nucleus of one of the Acetabularia species and the Acetabularia crenulata onto the stem of the Acetabularia mediterranea, which had a different cap shape. And in this experiment, he observed or he found that cap that grew in the transplanted cell resembled that of a Acetabularia crenulata, and he concluded that the nucleus influenced the development of the cap by transmitting factors or substances to the cytoplasm. Next, we will study about the enucleation experiment in Acetabularia, where the chloroplast has a role. To find out if the chloroplast, not the nucleus, can keep the cell working, especially for photosynthesis. Remove the base or rhizoid that contains the nucleus and keep the stalk and cap which have chloroplast in light to test if they can photosynthesize. Observe how long the cell survives without the nucleus. This is the experimental procedure, and in this diagram it's clearly shown. And the study of enucleation Acetabularia. Findings is it is a short-term survival. Chloroplast can do photosynthesis for a while on their own. It's a long-term failure. Without the nucleus, the cell eventually stops working because chloroplast needed help from the nuclear genes. And in conclusion, chloroplast can work independently for a short time, but for a long-term survival, the nucleus is needed to support the chloroplast. So without the nucleus, also a cell can photosynthesize with the help of a chloroplast, but it's not for a long-term survival. Next, we will study about the enucleation experiment in Acetabularia, where the chloroplast has a role. To find out if the chloroplast, not the nucleus, can keep the cell working, especially for photosynthesis. remove the base or rhizoid that contains the nucleus, and keep the stalk and cap which have chloroplasts in life to test if they can photosynthesize. Observe how long the cell survives without the nucleus. This is the experimental procedure, and in this diagram it's clearly shown. And the study of enucleation as a tabularia findings is it is a short-term survival. Chloroplasts can do photosynthesis for a while on their own. It's a long-term failure. Without the nucleus, the cell eventually stops working because chloroplasts needed help from the nuclear genes. And in conclusion, chloroplast can work independently for a short time, but for a long-term survival, the nucleus is needed to support the chloroplast. So without the nucleus, also a cell can photosynthesize with the help of a chloroplast, but it's not for a long-term survival. Next experiment is on sea urchin eggs to understand the role of nucleus in the development by removing it from the fertilized sea urchin eggs and observing the effects of the development. Procedure: selection of sea urchin eggs. Fertilized eggs of sea urchins are selected, and the enucleation process are done by microsurgical techniques. And as a control experiment, normal fertilized eggs with an intact nuclei are observed for comparison. The findings of this experiment is enucleated eggs. Enucleated eggs undergo early cleavage due to the cytoplasm resources like mRNA and proteins, and the development stops after early division. The egg cannot proceed to gastrulation or form a complete embryo. As in control experiment, normal eggs with nuclei will undergo the complete development, where it formed cleavage after follows gastrulation, formation of tissues and organs, and full embryo development, and long term it's a failure. So as a comparison, enucleated eggs can divide initially but fail to form a full organisms or larva due to lack of the nuclear control over the gene expression and later developmental stages. As in control experiment or control eggs, continue to develop a complete organism, showing that the nucleus directs the development, regulates the gene expression, and controls the later stages like differentiation and tissue formation. As a conclusion, nucleus is essential, and nucleus is needed for the long-term development, gene regulation, and the formation of tissue and organs. And also cytoplasm has a limited role in this, while cytoplasm supports early cleavage and it cannot sustain the development or differentiation without the nucleus. So coming to conclusion, we know that these are the experiments which show the nucleus is important for the survival of a cell. So nucleus is the control center of the cell, regulating the gene expression and development, and it is essential for cell differentiation and ensuring the specialized functions in organisms. Enucleation experiment shows that without the nucleus, cell can survive temporarily but fail in long-term growth and regeneration. As a tabularia, nuclear transplantation confirmed that species-specific traits are controlled by the nucleus. Sea urchin enucleation studies demonstrated that while cytoplasmic factors support the early development, nuclear control is required for full organism formation. Overall, the nucleus is critical for gene regulation, tissue formation, and substance life beyond the initial cellular processes. Thank you.